For activity 2, we will be completing our first Ed Puzzle activity, which you've probably already figured out since you're sitting in Ed Puzzle right now. And so the way this is going to work is that you're just going to watch this recorded lecture, and it will stop and pause and ask you questions throughout. It may ask you to answer a multiple choice question that's checking your understanding and your attention to the lecture, or it may ask you an open-ended question that wants you to provide an explanation or an example. And so remember that you will not automatically get a score for this activity in Canvas. I will have to go back next week and look at your look at the participation in the activity and then assign you a grade. So what we're going to talk about in this lecture is all relevant to the reading that you did in Chapter 3. And so we're going to broadly talk about how we started with nature versus nurture, and then really how we look at the interactions between nature versus nurture now. And so this is hopefully a debate that you've heard about before. It was the debate that essentially started developmental psychology, and where original theorists thought that either nature or nurture was responsible for the end product of human development. And nature in this analogy would be your heredity or your DNA or your genes. And so those are all the things that you are born with. And so this idea says that how you turn out to be after development is essentially determined at birth, or more so determined at conception, and that the environment doesn't really matter. Whereas nurture is on the other side of this, and nurture refers to everything that you experience after conception, and so that can be prenatally, since you're in an environment prenatally, or also all of the experiences you have after you're born. So people used to argue all the time that nature is more important or nurture is more important, but we know now after our decades of research that we can't look and say what's more important than the other. Really, they're equally important because they both interact with each other. And so the more important analogy would be, what's more important for calculating the area of a triangle, the length or the width? Well, you have to have both of them to calculate the area of a triangle, and one is not necessarily more important than the other. Also, the value of one, the length, will affect how the value of the other, the width, is multiplied. And so you cannot parse them apart. They're equally important and they're impossible to separate. And so really, if you want to come up with an image for how nature and nurture affect development on their own, we have a very hard time parsing them apart. Right at conception, when that embryo is first formed, you have some factors that have to do with heredity, the genes you just got, and then you also have factors in your environment. And at that point, it'll be the prenatal environment. But as soon as both of those things exist, they start interacting and swirling around each other and bouncing off of each other and causing cascading effects in one another. And then by the time you get to be a little baby, it's very, very hard to parse apart what might be causing what. And then even more difficult to parse apart what might be causing what once we get further on to development down here at the end of this funnel to looking at an adolescent or an adult. So we try to do experimental studies to isolate the effects of one factor so we can understand how it affects development, but experiments are very hard to perform in pure experiments are hard to perform in developmental psychology because you can't take someone out of their environment to isolate just one principle. And so really what we do is we collect data and we get a lot of correlations and use those correlations to build models and then make predictions about what might come. And so that's more like how a meteorologist gets all of the data on lots of variables on the weather and the environment and what's happened in the past, and they run them all through advanced statistical analysis to make a prediction. The important thing is that when we look at developmental psychology is that we have to have a lot of correlations. We're looking at a lot of relationships among variables and individuals. Those correlations have to add together so that we can really come up with some predictions. Taking one correlation out, as you will often see in the media or some things that you read online or headlines, is very misleading. Because although intuitively it might seem like two things that are associated in a correlation might cause each other. They might, and that might warrant more investigation for us to figure that out with a semi-experimental study or a longitudinal study. But sometimes things that are correlated are just random. Correlation does not indicate cause and effect at all, even though it might seem intuitive. But here's an example of an illusory correlation. So just two things that randomly go up and down together. Okay, so a correlation would say that, you know, both these things increase the same rate, or as one increases, the other decreases at the same rate. But sometimes these things just happen randomly. So this correlation that we're staring at is a very actual close correlation. You can see that the number of people who drowned by falling in the pool has just happened to correlate with the number of films Nicolas Cage appeared in. And so if we misinterpreted this, we might say, oh man, watching Nicolas Cage movies causes you to drown in a pool. People can't watch Nicolas Cage movies anymore. Now, this example is meant to sound ridiculous, although this is a real correlation, but it goes to show that this reasoning is just as faulty as reasoning that we might use to incorrectly explain cause and effect relationships with other correlations that might seem more intuitive. For instance, the correlations between vaccination rates and autism rates are highly correlated. As the number of vaccinated children has gone up, the number of autism occurrences have gone up as well. But again, that's just a correlation, and that might not be random, but sometimes correlations are caused by a third factor that was not looked at or that isn't mentioned in the correlation. And so we see similar rationale back here in the late 40s and the early 50s with polio, when polio was a big scare, and it was killing a lot of people and making a lot of people very ill. Well, looking at some of the data, they realized that polio was highly correlated with the rates in ice cream sales. And so you'll see these things go up right in line, polio rates and ice cream sales. And so after misinterpreting that correlation to be cause and effect, this was advertised and people stopped buying ice cream in order to avoid getting polio because they thought that ice cream caused polio since the rates went up. Similar scares were made about swimming. We saw very similar correlations between swimming, how often and how many people were swimming, and the rates of polio as well. So the correlations were very close, but really there was a third factor that was not mentioned in any of these correlations, and so they were taken out of context. That third factor was the temperature. And so polio was a seasonal illness. It increases and is spread through warm temperatures. And so as the temperatures increased and it got in summer, polio was more rampant because it was being spread more easily and the virus was stronger in that weather. And also higher temperatures also cause increases in swimming and increases in ice cream sales. And so some of these things might make sense intuitively, but we cannot come up with cause and effect relationships from correlations. If we have a lot of correlations together, looking at variables across a lot of people and a lot of correlations within those people, we can put those all together to make some predictions about what might happen next, but we certainly can't say one thing causes the other. And so this makes studying developmental psychology really difficult. Because a lot of things are interactions, again, between almost everything is the interaction between nature and nurture. So we strive to understand what parts of nature and what parts of nurture contribute and how much. But is there anything that's just genetically determined that we don't have to jump through all these hoops and think so complexly about? Is anything determined from birth? Well, very few things are determined from birth, but there are some. And there's even exceptions to these rules. But some things like physical features that are only dependent upon two genes that are received from both the parents, like earlobes. I don't know if you knew, but everyone has very distinct earlobes. And so your earlobe shape is actually determined from your genetics from your parents and isn't really affected by the environment at all. Eye color, blood type, and certain rare genetic diseases like sickle cell are automatically determined from birth and don't really depend on the environment at all. They're just the genes that you received. but everything else is some level of interaction between the genes and the environment. And so understanding these types of interactions and what different types of interactions there are will really help us to understand how we get this end product that might be one individual person or an individual trait in that person, an ability within that person. And so we're going to start at the lowest level of these most simple interactions of how nature and nurture interact to produce some type of developmental outcome. So the most basic level is critical or sensitive periods. And so these are a time when particular developments are supposed to happen, but they require the appropriate environment. And so we're pre-wired to develop some things at certain times. And during that period that we're pre-wired to develop those things, that particular area of our brain or body will be very, very plastic, which means it's very moldable and able to be developed and changed. All it needs is the basic and correct environment to do so. And so we're pre-wired to make it happen. We just have to make sure that the basic needs are met. And so one example of this, we see these starting even prenatally. There's quite a few prenatal sensitive periods that are occurring. One important one, starting in the 5th month, is the sexual differentiation of the brain. And so once the sex of the baby is determined, that's pre-wired, that will pre-wire the brain to develop in a certain way to be in line with whatever that sex is. But if the environment isn't right, if the hormone levels in the environment are wrong in the prenatal environment, or a toxin is presented in the prenatal environment, then that sensitive period might react to it, and it might not develop as it was pre-wired to develop. And so we can see some things going awry there. After birth, there's a lot of critical and sensitive periods where essentially the brain is pre-wired to develop a certain thing, and it just needs to be in the basic environment that to develop those things. And so if we look at this image here that just popped up, you see, we'll start here with the orange. The orange is sensory pathway, so that's developing vision and hearing. And so the higher it is up here is how plastic the brain is. And so that's the critical period for developing it. So right at birth, you'll see that the brain is very, very plastic and is ready to take in visual stimuli and develop the ability to see everything. Babies can't see very well at birth. They can only see very close to their eyes, and they can only really determine stark contrast between dark and light. And so they can see the difference between the white of an eye and a pupil. So they're able to look at eyes somewhat. They're also able to see the light and the dark contrast between a breast and a nipple. And so that's why when they're born, essentially the only two things they need to see. But afterwards, they have to be exposed to visual stimuli, nothing fancy, just the basic stuff, just literally seeing things to be able to develop, for the brain to develop to be able to see things in the future. So it's very plastic here, and then it goes down the brain, and that area is less plastic. So say, for instance, you had, blindfolded a baby up and throughout its first year, not that anyone would ever do this or that we could do it experimentally, but after that, the baby wouldn't really be able to develop vision normally since the critical period for developing vision has passed. We also see another one with language. So language peaks a little bit after vision, so more towards the end of the first year. And so all the baby has to hear during this critical period where the hump is its native language. And we'll talk more about this when we get to language development and bilingual babies. But they have to hear it, that language at this point, otherwise it will be much more harder, much more difficult to develop that language later on. Now we see the critical period for higher cognitive functioning is happening a little later. So really babies just have to sit there and hear and see things. But this is an interaction between nature and nurture, because if that environment isn't there, if it's not exposed to visual stimuli or to its native language, that baby won't be able to develop the ability. So that's just basic level. We don't have to do anything fancy with our babies. They just have to have, in this first year, just the basic stimuli. There can't be any missing components in the world This is a similar, showing similar concepts. So we, of course, here at 0, we're starting with binocular vision. So the darker it is, the more plastic the brain is there. So in the first year, it's very plastic, and it's really just looking to see things and develop the brain that way. See emotional control, which we'll talk about next week with attachment. The babies are very plastic, that part of their brain, at the end of the first year and throughout to the 3rd year. And this is where attachment with the caregiver can really determine how the brain develops and how that person will be able to respond and cope with stress and emotions later on in life. See peer social skills, not starting until 3 here, but this is a very critical period where children learn the basics of cooperating with others, language, symbols, and then once we get to this higher level cognitive functioning, this doesn't start until later on. So this is really the lowest level of these interactions. The environment isn't affecting these things so much, just needs to have the basic components of the environment for these things to develop naturally as they were pre-wired. These are the natural, what we would call typically developing features or abilities of a person. But what about abnormal development? How much does genetics or the environment contribute to, say, the development of a disorder? Well, no disorder that we know of, some diseases, but not very many disorders are entirely determined by either genes or the environment. Really, we aim to determine how much genes or how much the environment contributes to a given disorder. And really, since we can't run experiments on this, the best tool we have for understanding this that you read about in your book is looking at twins, because we know that the twins have a certain amount of overlap in their genetics, depending on the type of twin. And then also we can look at twins that were raised together or twins that were raised apart to see how the environment affected the development of something in particular. And so these concordance rates essentially just determine the percent of variability and the tendency to have a trait that's due to genes. And so identical twins, of course, are going to have 100% gene overlap. They come from the same embryo. And then fraternal twins are going to have some overlap, but they really have the same amount of overlap as siblings. They are essentially our siblings. They just shared the womb together and were created at the same time. And so looking at the differences between identical twins and fraternal twins can give us some insight into how much a disorder or a particular trait that developed is hereditary. So let's look at a specific example in a chart, and we can use autism, which has a lot of research going into it right now and is a pretty hot topic in developmental psychology, even in the lay media. So on this graph right here, we have the concordance rates. So this would say the concordance rate is the likelihood that if one twin or sibling has it, that the other one will also have it, okay? So you see, this is the new model for the concordance rate for autism spectrum disorder. Just like any other disorder we have, there is not a 100% concordance rate. So even identical twins aren't necessarily both guaranteed to have it. But there is a very high concordance rate with identical twins. There's a 75% chance that if one identical twin has it, the other one will as well. Then we see the next comparison here with fraternal twins. And so remember that fraternal twins have the same amount of genetic overlap as a regular sibling, okay? So the difference between a fraternal twin and an identical twin here, they are both have the same prenatal environment. So the only difference between them is that identical twins have more genetic overlap. So the difference between the identical twins here and the fraternal twins, this about 25% here, is that we can say genetics has at least 25% to do with being born with autism spectrum disorder. Now, the difference between fraternal twins and regular siblings here, remember that fraternal twins and siblings have the same amount of genetic overlap, okay? The difference between them is that fraternal twins share a prenatal environment. And so this difference here, we can see of almost over 25% is what we think is the effect of the prenatal environment. So something happening in the womb is causing the marked increase in rates for fraternal twins than siblings, okay? And so we can use these types of comparisons to look at how much genetics versus the prenatal environment versus the regular environment here can contribute to any known disorder when we look at these concordance rates over large groups of twins. We use this type of research on concordance rates to also look at different types of disorders. So take schizophrenia, which has a very large genetic component to it. This is the same type of thing, but just illustrated in a different way. We see that there's a 50% concordance rate with identical twins, which is actually 25% less than autism. So if one identical twin develops schizophrenia, the other one has a 50% chance of developing it as well. You see the difference between identical twins and fraternal twins. Again, that difference is largely due to genetics because they both share the same prenatal environment. So we see there's a very big genetic component that comes with schizophrenia here. But we need to think about why there's only a 50% chance of identical twins. Where is that 50% of the environment coming from? If it has half of it is genetically based, then how is the environment affecting it at all? Why might one twin develop it and the other one not develop it if they have exactly the same genes? Well, we can use a broad concept, has a lot of different names, but the one I like the best because it's easiest to remember and nice to visualize is this kindling theory. And so what is kindling? Kindling are the little sticks that you use to start a fire. Without the kindling, you cannot start a fire. And without the flame, you cannot light the kindling. And so here we would think of the kindling, this actual wood, as the genes. And so you might be born with these genes that predispose you to developing a certain disorder, like autism or schizophrenia. But unless something comes and lights and turns that disorder on, unless something comes and sets that kindling on fire or manifests that disorder, you may not develop it. And so you have to be born with the kindling, and then something has to come along in the environment and set it on fire. So this is the combination of having a genetic vulnerability, of being born with certain genes, and then having the environment that helps that manifest. And so specifically for schizophrenia, this is talked about as the stress vulnerability model, but you don't need to worry about that name. Really, this is just going to show that here's the vulnerability, the different levels of the genetic predisposition. So someone that has a low genetic predisposition, here's the environment. It doesn't even matter if their environment really encourages the development of that. They will never develop it because they aren't born with the genes, right? It's kind of like if you want to think of someone with cancer, I'm sure everyone knows of at least one person that has smoked their entire life and hasn't got lung cancer, right? They may just not have the genes that help predispose them to developing that. On the other hand, you might know someone that either didn't smoke at all or only smoked a little bit and developed it. And so that's the difference between the vulnerability here. And so if you are born with the genes that predispose you to developing that disorder, if you don't have an environment that encourages the development of that, you may never develop it. But if you have the environment that does trigger or encourage the development of that predisposition, then you will have it manifest. And so really it's a specific combination of how your genetics predispose you and then how the environment encourages the manifestation of whatever that is. But how does that happen? How would an environment change whether a gene manifests or not? Genes might mutate and randomly develop or change over time, but not really. That's rare, especially after birth. But what happens is that our environment constantly changes the way our genes are expressed. And so that's what we're going to focus on in activity 3 as well. So this is called epigenetics. Have you ever heard of epigenetics before? Well, it literally translates as above the genome. And so your genome are your genes. You are born with your genes, and we cannot change the genes that you've been given. But what epigenetics looks like are the things that sit on top of your genes, on top of your DNA, and change whether it is expressed or not. So it doesn't change your genetic code, it just changes how that genetic code is expressed. And so your environment can actually have very large effects on your epigenetics. So your environment can turn off, on or off certain genes. And what we mean by on or off is it can express a certain gene and cause that something to manifest. So say it can express the schizophrenic gene and make that disorder develop later on. Or environmental factors can turn that gene off. So even though you have it, it isn't active and awake and doesn't actually manifest into the disorder. So this might make a little bit more sense if we look at what DNA and epigenetics actually look like. So here is our DNA, okay? All of these little things within it are genes. And so your DNA is wound up, but it can be wound up tightly or it can be wound up loosely. So let's actually go to a different visual that will really give you a better idea. I have to allow my flash player. Okay. So you're born with these genes. But if a gene is coiled up very, very tightly, I mean, if your DNA is coiled up very, very tightly, then whatever gene is on the inside will not be expressed. It's kind of hidden and never goes active. And so you see here that these yellow strands of your DNA are wrapped around histones. And so these histones and these other little dots, those are your epigenetic markers that are controlling how much your DNA is wound up or how much it is kind of loosely, flattened out and expressed. And so all of the genes on this yellow piece of DNA you see here, if we unwind it, you can see how more of this DNA is exposed now. And so any of the genes that are on this exposed part of the DNA are now going to be expressed and active. Whereas if we go in the opposite direction and wind it all the way up, we can see that none of those genes are expressed now. They're all dormant and rolled around those histones. And so these histones and the little markers are telling your DNA what to do. And your environment directly affects these, so that's what we call epigenetics. And your environment directly affects these histones and these methyl groups. And so what your DNA does is largely affected by your environment. What is on these yellow DNA threads does not change at all. It's just how tightly they are wound. Okay, so it's very important to make the distinction that your genes don't change, but how they are expressed may change depending on your environment. And so let's go back to the PowerPoint now and talk about that just a bit more. So if you want to think of it as an analogy, you can say genetics is the loaded gun, or genetics is the kindling, but epigenetics pulls the trigger, or epigenetics lights that kindling on fire. And so you have to have that gene expressed for it to manifest in real life. And so your epigenome and what genes are expressed or are dormant and turned off can change throughout your lifespan, and but only to a certain point. Okay, you're not going to be constantly unraveling and wrapping up your DNA throughout your life, although some factors that we'll talk about in a minute can cause that to happen. Epigenetics are particularly sensitive during pregnancy and puberty, so plastic periods of development. Okay, so the mother's epigenetics are very easily changed during pregnancy, and then the baby's epigenetics are easily changed during pregnancy or puberty. And so these are critical periods for either turning off genes or turning on genes. And so what can cause an epigenetic change? What can cause DNA to tightly ravel or to unravel and turn genes on and off? The most important thing is diet. For activity 3, you'll watch a TED Talk titled "You Are What Your Grandmother Ate," because your diet essentially has methyl groups in it. And so remember, those methyl groups were those little dots floating around that were telling the histones what to do. So they're telling the histones to either wrap that DNA up or to unwrap the DNA. And your diet might be full of methyls, or it can be lacking methyls. So the more methyl groups you eat, the more your DNA is going to wrap up. And so very leafy greens that contain folate, which are a methyl group, will put a bunch of methyls in your bloodstream, and it will tell the DNA to wrap up tightly, which would turn off any genes you have for possibly some disorders or undesirable traits. So your diet is very important. The way that we discovered epigenetics was actually by looking at the diet in mice. And so we had this breed of mice, this yellow one right here, that were these fat blonde mice. And they were born, the mothers were fat and obese, they were born fat, and then the babies, since they were born that way, ended up dying very early. And so they were inheriting this agouti gene, which is what caused the mothers to be blonde and to be obese. But what happened when they were studying, and they were actually, of course, as it happens with any type of scientific research most of the time, they were studying something else, and they found this as a byproduct. But as they were changing the mother's diet, they added a lot of methyls to the mother's diet, and they ended up having a litter of mice that weren't expressing the agouti gene. They had thin, healthy brown mice, even though the mothers had this agouti gene that caused them to be blonde and overweight. So what the methyl groups did when they added them to these mothers' diets is it caused the mother's histones to wind up that agouti gene and turn the agouti gene off. And so the babies were born with that agouti gene turned off. It did not manifest. And so the babies were not born expressing the agouti gene. And so just by changing the mother's diet, making it something high in methyl groups, they were able to turn off that very dangerous gene. And we see very similar things in humans as well. So your mother's diet during pregnancy and your diet as an infant can affect your epigenome in ways that stick with you into adulthood. Animal studies have shown that a diet with too little methyl-donating folate or choline, which are all those little dots that tell your histones what to do, before and just after birth causes certain regions of the genome to be under-methylated for life, which means that that gene will be kind of stretched open and exposed throughout the lifespan. So we see a very sensitive period here, prenatally and directly perinatally. For adults too, a methyl-deficient diet leads to a decrease in DNA methylation, But these changes are more subtle and they're reversible when methyl is added back to the diet. And so throughout your lifespan, you can be, depending on your diet, you can be raveling, tightening the ravel or loosening the ravel on your DNA and expressing or not expressing certain genes. But however your epigenetics are prenatally, you will actually pass those on to your child. So just like the mice in the original example, you can pass on a gene that's either expressed or not expressed. What else can cause epigenetic changes? Well, your diet, as we just talked about, alcohol, drugs, and smoking can affect the methyls and the expression of DNA and the shape of your DNA. Stress and trauma can affect it, sleep, and environmental pollutants. And so epigenetics is a relatively new concept that we've only known about for 20 years, but it can explain a lot of the interactions we see between genes and the environment. So someone having the gene for something and then the disparity on whether it manifests or doesn't manifest. And again, you pass this epigenome down through generations. And so if your grandmother never ate any greens, that DNA is really stretched out and open, and all of those genes are active. When you get that DNA and that gene will be open and active. Or vice versa. You can pass it on if you have a lot of methyls in your diet, as the DNA is tightly wound. And so we're only really beginning to realize how important the diet is, especially prenatally. But luckily, pregnant women already have all sorts of rules about what they're supposed to eat and not eat. But maybe add to that list now to eat a lot of methyl-rich folate greens to help wind up the DNA and turn off any less desirable genes before we pass them on. And so when we're looking at humans over the lifespan, we can actually see some really interesting things from twins about how the environment can affect your epigenetics over the lifespan. And so this first picture here is 3-year-old twins. And so these twins would have been born with the same epigenetics. These are identical twins. So they're born with the same DNA and they're born with the same epigenetics, since they're in the same prenatal environment, right? And so when they're only 3, their epigenetics are still looking very similar. This yellow here shows where their methyl groups and histones are looking very similar and looking identical, okay? So the yellow places are where their epigenetics are the same at 3 years old. But looking at the same pair of identical twins at 50 years old, you can see that their different lifestyles and the things that they've experienced and their diets have made their epigenetics very different. And so these red areas here are where their histones and methyl groups are very different and their DNA is expressed very differently. So again, the same exact DNA, but here in the red, there are differences on how that DNA is wound up or unwound. And so we see that the environment can have a very, very large impact on how the DNA that you were born with is expressed. And so we'll get more into epigenetics in activity 3, but for now, we're going to move into some of the less genetic interactions, and we're going to get into Bronfenbrenner's ecological system, because this class is going to focus on how the social environment can affect development. And so you probably have seen a Bronfenbrenner ecological system in a previous psychology class if you've ever had one. And so the idea is that there's a lot of different layers of how the environment can affect us. And these systems, we view the development as a byproduct of the transactions between these different levels. Okay, so there's lots of different levels, and let's go over each one in just a minute. And the thing is that the main idea is that these are bidirectional. So each level can affect each other, and then that will have a subsequent effect on the developing person. So here is the ecological system. So in the middle here, you have the individual. Right outside the individual is the first, you might say, most important layer of their environment. So it's everything that they're directly exposed to, their microsystem. And so a microsystem would include anything or anyone that the individual has direct interactions with. So you have direct interactions with family, with your friends, with your school or church. So anything that you're actually interacting with is your microsystem. So can you think of any other examples other than family, friends, school, and church of something that might be in a microsystem that you directly interact with? The next layer is the mesosystem. And so the mesosystem is interactions between those things in your microsystem. And so that would be, you know, an example of your parents interacting with your teachers, or your parents interacting with your friends, or your friends interacting in your church. And so these are just the interactions among things within the system. So could you think of any other examples? The next layer is the exosystem. And so that's a little further out. And these are things that you indirectly interact with. And so your parent's job is a good example of this. So you don't, as a child, interact directly with your parent's job, but their job can have indirect and significant impacts on you. And so how much money your parents make at that job can have a very significant effect on this individual. How stressed out your parent is at the job, how often the parent has to work. And so these things can indirectly affect development, even though the individual isn't actually directly interacting with the job. They're not at work with the parent, although in some cases they might be. Going up another level, we have the macro, culture and society. So that's the culture that you live in, the beliefs that you have, the norms within the culture, the laws that you have to obey within that culture can have pretty significant effects on this individual child, right? And so can you think of any maybe belief within the macro system right now in our culture that's affecting the development of a child, as an example? So chrono changes is the last layer, and this is things in the broader culture that change over time. And so these are changes to the system, okay? So the death of a loved one would be a change because it's changing something within any of these layers. So this is a really complex and kind of broad system here that we won't focus on too much. But what's most important here is going to be this microsystem, the direct interactions, the mesosystem, how those direct interactions interact with each other, and the other systems, exosystem and macrosystem. And so, like we said, it's impossible to take the individual out of the environment to assess the effect of one thing or another. And that's because all of these different layers of the environment are interacting with each other. And it's very hard to parse one apart individually. So even within this model, it explains things like why two siblings in the same home won't have the same experience. Just because two kids grew up together, right, does not mean that they had the same experiences, or does not mean that they had necessarily the same environment growing up, although it will be more similar to kids that aren't siblings, right? But each child has different genetics, and they might have a different inherent temperament or personality, as we'll talk about in a second. And this will then affect how he's treated by others. And so all of these layers of this ecological system, the genetics of the individual, and then all of the environmental layers are interacting with each other and causing change in one another. If you want to think back to that tornado, everything's bouncing off of one another and causing changes in the other. So there's three specific types of relationships that we'll talk about relevant to that. And so next week, we're going to talk more about temperament and personality. But for now, all you need to know is it's pretty stable. Your temperament is the same as personality. We use temperament to describe it in babies and young children, and then we use personality to describe it later on. But essentially, they're the same concept. And it's pretty stable, except when people are going through puberty and there's all sorts of things affecting it. But we believe it to be largely inherent. And so for babies, there's three general categories for temperament that we have. An easygoing baby, a difficult baby, or a slow warm-up baby, which would be more like a shy baby. And then, another third of babies don't really fit into any of these categories. But essentially for these broad types of temperaments and personalities, we see that adults will be the same as they were in babies, although who's to say what's going to happen during adolescence? We'll talk about that more later on in this semester. So the 5 main personality traits that we think about when we think about temperament or personality are openness to experience, conscientiousness, so how thoughtful someone is, extroversion or introversion, agreeableness, which is how easygoing they are, and neuroticism, which is how many negative emotions someone typically has. Most often, our personalities correlate with our environment. So our personalities match with what's going on in our environment. We consider this, as you read about in your book, to be goodness of fit. And the reason that happens is because there can be 3 different types of interactions between someone's genes and their environment that end up making them similar and fit together. So a passive type of gene-environment correlation would be essentially you got your personality trait from your parents, and then your parents choose what environment that they put you in. And so if you have a very extroverted parent, they pass those genes on to the child. That child is also very extroverted. They got that gene from the parent. And so that child correlates well with their environment. They're an extroverted child in an environment that requires a lot of extroversion simply because they got it from their parents and their parents are choosing that environment. So really it's the parents here providing the environment, and the kids are more likely to be like the parents. The next one is evocative correlation. So this is where the child is causing changes to the environment. So when an individual's genetically predisposed behavior evokes a response in the environment. So a great thing to help make sense of this is that children are like mountains that create their own weather. So if you're already dealing with a very irritable child, the way that you interact with that child is going to be different than how you would interact with a very passive or slow to warm up child, right? Because the environment is reacting to that child. And so they are causing a change in their environment, and that change makes the environment more in line with their temperament. Now, this isn't always good. It could be that a very hostile or irritable child is causing their environment to be more hostile and irritable. Or it could be vice versa, that a very calm and inhibited child is causing their environment to be more calm and inhibited because it's reacting to them and trying to accommodate them. An active one, this is the third and last type of correlation, is where children would more actively seek out environments that fit their personality. So an extroverted child might seek out extroverted activities, or an inhibited child might seek out games or activities that are quieter and require less social participation. And so this is them finding the environments that suit their personality. So the differences between these might seem somewhat confusing, but it's important to be able to parse apart how the environment the child is placed in might line up with them well because their parents placed them there, or how the child can cause changes to their environment that make the environment more in line with them, or how the child might actively seek out an environment that's more in line with them. So coming up with examples of each can help us parse apart and determine the difference. So let's say I have a child with a high verbal ability and a highly verbally stimulating environment. So here we see a gene environment correlation. You have a very smart child with a high vocabulary, and they're in an environment that is really stimulating that environment and dealing with them a lot verbally. So passively, the child might have inherited a high intelligence from their parents, right? So they have high verbal ability naturally. And so you have a highly verbal child in a highly verbal house. And that's just because the parents have a highly verbal house, and then they then pass that gene and environment onto the child. What would an example of an evocative correlation be here? How might this correlation be caused by an evocative relationship? Remembering that an evocative relationship is where the child causes the environment to react to them in a specific way. Well, perhaps since the parents noticed that the child had a very high verbal ability, now they're giving them a lot of reading material. So the environment is reacting to the child's temperament. The child is causing the reaction in their environment, and now you have a correlation between the child's temperament and their environment. What about an active relationship? How would this correlation be explained by the child seeking out this type of environment? Well, there's a typo here, but Perhaps the child enjoys reading and asks to go to the library to rent books. So they're seeking out an environment that's more in line with their temperament. Let's say we have a child with an antisocial personality, so they're kind of mean and aggressive and hard to get along with and highly irritable. And they're living in an abusive environment. So you have a correlation here between the child's temperament and the environment that they're living in. How might the child might have passively inherited this environment that's just like their temperament? Well, the parent might have these antisocial genes. Antisocial in psychology doesn't mean that they don't like to socialize with people. It means that they go against norms and break rules. And so might have a violent antisocial parent, and they pass those genes on to the child, and then leaves them in a antisocial environment where they don't supervise them. And so again, the child got it from their parents. Now they're sitting in a situation, in an environment that the parents set up. How might this be explained by an evocative relationship? How might the child cause the environment to be abusive? Not that a child would ever cause an environment to be abusive, but well, the child could act violently and elicit violent behavior from the parents. And so oftentimes you'll see or hear about violent children being spanked as a punishment for their violent tendencies. And so this would be an evocative example. A child is hitting another child, and then the parent comes and takes and spanks that child for being aggressive in the first place. And so they're causing an aggressive reaction by acting aggressively. And so we have a correlation here and a goodness of fit. And those two things being similar and correlated would cause that type of personality to persist. And so spanking or using other types of corporal punishment to punish aggressive behavior simply just makes the environment fit better with that current temperament, and it makes the child more likely to maintain that temperament. How might the child actively seek out the same environment? Well, the child could act out intentionally to receive negative attention from the parents. And so all of these types of correlations, having goodness of fit between the temperament and the environment, makes that temperament more likely to not change. If you wanted the temperament to change, you need to make sure that the environment does not fit in with the temperament. So we wouldn't want to aggressively punish a child for being aggressive. We wouldn't want to make these things line up. So let's try, you can do all three for this one. So we have a cuddly, introverted child who is constantly held. So a needy kid, they're constantly being held by parents and families and friends and teachers. So how might this have passively occurred? How might this correlation passively develop? Well, an introverted parent could have passed on an introverted temperament to the child, and the parent just naturally want to cuddle and coddle the child for comfort since they're introverted. And so the child just passively inherited both the genes and the environment that fit it because of their parents. What about an evocative relationship here? How might the child cause the environment to be one where people are constantly holding them? Well, the child could cry whenever they're not being held, right? So the parents or teachers or whoever might go and spend extra attention on that needy child because it's obvious the child's temperament needs that, right? Active would be very similar in this situation, but see if you can come up with a different example for how a child might actively seek out this type of environment. Sure, they can just go up and ask, say, hold me, hold me. So they're actively seeking out an environment or the types of games or stuffed animals that's going to fit in with their personality. And so how these three types of correlations between the temperament and environment affect goodness of fit over time can change depending on how old the child is. And so the active genotype environmental interactions, you see, children can't really actively seek out environments that fit in with their personality when they're little, because they're not choosing their environments, right? Their parents are putting them in the environments when they're little. And so you see that passive environmental and genotype interactions are really coming more to play here, since the parents are the ones that are picking and providing the environment. As the child gets older, you see that the active interactions are increasing, that the children are more able to seek out environments that are fitting with their temperament and their personality as they gain more autonomy. Whereas these passive ones, where the parents during adolescence are really not choosing the environments quite as much, dissipate a little bit. Evocative environmental interactions are always occurring, and they always have a similar effect on the child. And so if we either want to keep a good temperament or an easy temperament the same, or if we want to change a difficult temperament, that's where really the environment will come most into play, because we have a little bit more control over that. So if there's a temperament that is not ideal, you want to try to not react to the child in a way where your reaction or the environment is going to correlate with how they're already acting. And again, we think this is all very stable temperament and personality, but obviously it can change a little bit, especially depending on those evocative correlations and how much they're provided. So will early characteristics carry on throughout life? Most of the time, but not necessarily. There is some room for those evocative correlations to make an impact on temperament. Again, these negative correlations are the ones that would change temperament, because they're reducing the goodness of it. So we covered about 3 different levels of interactions between the genes and the environment here. Starting at the prenatally, they're already starting to interact with sensitive periods through hormone levels and epigenetics and the environment prenatally. And then from there, they continue to wind together, interacting with each other. These social environments, all the different levels are interacting with each other. The physical environment, like diet, are also interacting with genes, epigenetics, and all of these social layers. And so it's really very hard to parse everything apart, but we do know how certain types of interactions are taking place. And I can't stop this.